Hello, and thank you for joining Cure Alzheimer's Fund today for this important presentation. I am Meg Smith, CEO of our organization, and I hope you will allow me just a few minutes to introduce CureAlz. We were founded with one mission: to fund research that will accelerate prevention, treatment, and cure for this disease. To that end, since 2004, we have deployed more than $200 million toward extraordinary research pursued by brilliant and determined scientists around the world. Our dedicated donors make all of this possible, people who share our drive and commitment to end this disease and its terrible impact on patients, families, and our communities. We are grateful for their trust in us, and we are determined to honor it by finding and funding the vital research that is the only way to achieve progress. Our board of directors and trustees hold us accountable to our mission. They and certain other supporters cover all of our operational overhead. This generosity and vision mean that our other wonderful donors know that 100% of their gift goes directly to our research program. At CureAlz, our only goal is to find solutions to help the Alzheimer's community. We do not pursue profits or financial returns. All of our scientific decisions are made by active researchers who themselves are at the forefront of discovery. Our research leadership group guides our scientific decision-making. They are experts in scientific specialties relevant to Alzheimer's who lead labs at top academic research institutions around the globe. They determine what investigations we will support, always ensuring we pursue the ideas that science and data tell us are the most likely to make a real difference for current and potential Alzheimer's patients. Thank you again for joining us to hear about the science and progress that matter in Alzheimer's. We hope you will continue to join us in our mission to end this disease.
I'd like to first start by sort of telling you a little bit about myself in terms of how I became interested in Alzheimer's disease research. And as you may have gathered from the fact that I'm associated with Boston Children's Hospital, you might be wondering why someone who studies brain development traditionally, and I am a developmental neurobiologist by training. Many years ago when I started my lab, I realized that some of the same ways that we sculpt the developing brain, which is really important for normal brain development, we sort of prune and remodel synapses. These are the connections between neurons. Even early days, we started to appreciate that some of the same mechanisms or ways by which this works may be actually quite relevant to Alzheimer's disease and also other diseases of the aged brain. And back then, I took a chance, without having the background in Alzheimer's disease or neurodegeneration, to, in parallel to our developmental work at Children's Hospital, I launched another side of my lab that was really dedicated to testing some of these, at that time, very out-of-the-box ideas in the context of Alzheimer's disease. And through the support of Cure Alzheimer's Fund and others that kind of really support bold ideas, new ideas, at a time where maybe I wouldn't have normally gotten funded through the NIH and through more traditional mechanisms, it gave me a chance at the very early stages of my lab and career to sort of launch some of these new programs and projects in the lab. And I have to say, if it hadn't been for that, I don't know that I would be here telling you about what we discovered. And not only has the work we've been doing on Alzheimer's disease been informative in terms of the biology, which of course, as a basic scientist, this is really important to understand how cells in the brain work and how these mechanisms work. What we're starting to now appreciate more and more is to translate and to try to, discover new therapies or even new biomarkers that would tell us whether someone is at more risk for developing some of these diseases. It's really important that one really understands how it all works, because that's how you target the right cell type at the right time or the right pathway. That's how you come up with new medicines. So what I want to tell you about today is a couple of examples of that. And I'm going to focus in particular on a group of cells, in particular one cell type called microglia. These are our resident immune cells in the brain, shown here. And on the right, you have sort of an example of these microglia. These are small little glial cells. So these aren't the neurons that you're probably more familiar with in the brain. These are cells that make up about 8 to 10% of the cells in our brain. And they're incredibly dynamic, as you can see from the different shapes that they have in the brain. But one of the reasons why, even decades ago, pathologists and others in the Alzheimer's disease field became interested in them is if you look in the brains of Alzheimer's disease patients, where there's a lot of the amyloid plaques, these are these toxic protein plaques that accumulate in the brains of Alzheimer's disease patients, it was observed early days by looking at some of those brain sections that the microglia surrounded, right, the plaques. And that raised a lot of questions back then because these were just snapshots. What are these cells doing around the plaques? Are they good? Are they bad? Are they helping to get rid of the plaques? Or are they in fact contributing in some way to the disease? And that is actually the theme of what I want to tell you about today. And I guess the sort of quick sort of take home of the lecture today is that it's not one black or white answer. They don't just do one thing. They do lots of things. And because of that, it is the reason why really delving into the research, the biology in terms of how they do these different things is really important. Because if you're going to target their good roles, you need to understand how to do that, properly, how to make them more efficient in the brain. And at the same time, if you find that there's some bad roles that they play in the brain, you want to specifically target those roles, but not the good roles. I know that's an overly simplistic way of thinking about it, but in fact, that is the challenge right now that lays before us because it's not so simple. So we really need to dig into the biology, and we need to develop new approaches with which to do that. And I'm going to give you a couple of examples of that today. And this is just another example of the microglia that are around. The purple here are the amyloid that builds up in the brains, not only of humans with Alzheimer's, but also animal models that we can generate that kind of mimic some of the same symptoms of Alzheimer's in the mouse brain. And that, of course, is more amenable to research. So we can actually study this over time, and we can start to ask, when do the microglia change? What do they do? Are they good? Are they bad? Especially in the context of amyloid and other Alzheimer's pathology. But I want to kind of zoom back out again and just tell you a little bit more about microglia. These are my favorite cells. So I'm going to kind of give you a little bit of sort of a tutorial in a way, but in a way that I hope gets you interested in them. So as I mentioned, these are microglia are about 8 to 10% of the cells in the brain. They're really beautiful. I'm of course biased, but this is a microglia with all its little fine processes. So they kind of tile the brain, okay? And we have ways that we can label them with fluorescent markers like this one, but we can also label the microglia in tissue from lots of different species. But we study them not only in a mouse, we can study them in the human brain, and we can also develop models where we can actually generate human stem cell models of microglia, and we can study them in a petri dish. And I'm going to give you a couple of examples how each of these approaches is really helping us to better understand their biology. So here they are. This is just one microglia. And what scientists, including ourselves, have noticed is when there's an insult or an injury or a change to the brain, they can really dramatically change their shape or their function. They become more like this guy. And, this was often termed the word activation. So they seem like they've sort of pulled in all those beautiful processes, and the red stuff that's inside of them is something that they've eaten. So they've engulfed it, right? Like think about Pac-Man or, sort of cells that can actually, eat something. These cells are incredibly good at engulfing. When you look at a cell like this, you say, well, wow, something bad happened. And while that's true, just by looking at them like this, it doesn't really tell you when they changed, which of course we really want to get to really early, like what's the first things that happen in the context of a disease like Alzheimer's disease, because often we're just getting snapshots, right? We want to know specifically how they contribute to Alzheimer's disease. And we want to really try to better understand, are all microglia created equal? Are they all doing the same thing? Or could they be kind of doing different things depending on the timing and the context? And I'm going to give away the last answer. And the answer is there are lots of different states of microglia, both good and bad. So one of the goals is to try to better understand some of those functions. So I always start with movies because to me, I got into this field by looking under a microscope and watching these cells, not just in fixed sort of snapshots of tissue like I just showed you, but we also have the ability in mice, not yet in people, but in mice, to be able to put a mouse's brain or a mouse under a microscope, and using ways to be able to label the microglia with green fluorescent proteins like you see here, we can actually watch the microglia in real time. So we can watch what they're doing in a living mouse, okay? And this is a great example of that, where we make a little injury to the brain, and I want to show you what they do when there's a damage to your brain. Now, this is sped up quite a bit, meaning it's, kind of a little bit faster than what would probably happen in real time. But I hope you can appreciate from this movie that they are doing a couple of things that are pretty amazing. They are sensing damage and they're moving their fine processes towards that damage. That damage is a laser injury that we made, but it could also be an amyloid plaque. They have ways of sensing when there's something bad or toxic happening in the brain. So I would say, well, that's a good thing. You really want them to respond and hone in on those things, and you really want to make sure they do it, really efficiently. At the same time, though, you might say, well, okay, if you want them to respond, you might want them to remove that sort of toxic protein or whatever bad damage thing that happened. But you wouldn't want them to suddenly become so activated that they start eating and engulfing things that they shouldn't be doing, like a healthy neuron, for example. And what happens as we age, it turns out in general, microglia's ability to do that kind of responding to the signal is a little bit dampened. It's less efficient. And also their ability to kind of regulate, the ability to kind of only kind of clear without becoming neuroinflammatory becomes sort of, imbalanced. And so one of the things we're kind of learning about is that, in a young brain, in a healthy brain, they're a little bit more dynamic and able to kind of go back to that good role that they do. But in the context of disease and injury, they can sort of go on overdrive and they can start to contribute to things like brain neuroinflammation, which also has a lot of detrimental effects in the brain. So we're really just trying to understand what regulates all these different states of microglia. So instead of just saying we just want to get rid of them all, or we just want to, activate or deactivate them all, that's a little bit too simplistic. We're really trying to better understand how to boost their, sort of good roles and decrease their detrimental roles, like their inflammatory roles in the brain. So another thing that we and others have noticed is even without injury, they're always sensing their environment. They're really, really good at sensing the environment, just like a macrophage, which is sort of their counterpart, would be doing in the immune system. And that brings us to another theme. I mentioned that these cells are our resident immune cells, and you might think, oh boy, I don't want to have immune cells in my brain. These are not those kinds of immune cells. These are cells that were born in they're from in our brains from the very earliest stages of development, and they stay in our brain our whole lifespan, most of them, which means they have a lot of normal functions in the brain. So in other words, this is not an example where, suddenly they come in from the peripheral immune system and sort of do all these things that I've just described. A lot of the microglia that I'm showing you have been in the brain the entire lifespan of a human or a mouse. And so we want to better understand what they normally do. And if you can understand their normal functions, that can better help us understand like how they go awry in disease. And it turns out they do a lot of important normal functions in the brain. So they are really important. They can have a major role in producing a lot of neuroinflammation, which you probably know is not such a good thing, although there are good forms of inflammatory proteins, but that's another story for another day. They're really good at clearing or engulfing, removing things you don't want in the brain, like amyloid and other debris, which can build up with time, and that can lead to inflammation. So you really want them to be able to get rid of the things that are sort of degenerating in the brain very quickly. But we also realized and studied a number of years ago, they also have a lot of other roles in the brain, especially during development, which is their role in sculpting, developing circuits and synapses. And I'm going to tell you about that in a minute. So if I could just sort of highlight the big picture here, they have a lot of normal roles, surveying the brain, promoting brain health, really like kind of sensing what's going on in the brain. So make sure that, that the brain stays in check and alerts other cells to make sure things stay in check. But when there's a challenge or when there's aging, it can shift into these other states. So one of the real kind of thrust of our research and the goals of our research is to try to better understand what shifts them into these different states. And ideally, we want to shift the detrimental states back to the state, and we want to promote their beneficial states. And that's really, in a nutshell, what we're trying to do. But the other thing I really want to highlight in the context of diseases like Alzheimer's disease is, this is a disease that happens over many, many years. And what we're really, as a field, trying to do is understand what's happening in this early, what we call preclinical stages of Alzheimer's, which is, there's no, indicators yet of whether someone's, starting to generate some of this pathology, or whether they're starting to show some of the earliest signs of sort of synapse loss or cognitive decline. In this window, I think this is a major gap in the field, is to try to understand what's happening at the very, very early stages of Alzheimer's disease. If we could understand that, in particular, if we could understand how microglia are changing across the entire spectrum of a disease like Alzheimer's disease, that would help us to better understand how to target the earliest changes. And I think that is something we're really trying to do. Now, in a human brain, we don't have the ability yet to go in and look into a human brain the way we can in a mouse. But I would say one huge goal for our research, and I think of the field, is to develop new biomarkers that would enable us to track microglia changes from the very, very earliest stages of Alzheimer's disease, 15 to 20 years before you start to develop cognitive decline, and to be able to monitor those changes in a living human over time. And one of the goals of trying to study this in a mouse and in all these models I'm going to tell you about today is that will give us better ways to develop new biomarkers. So like some of you may know, if you do a PET scan for amyloid, the toxic protein in the brain, we can detect that in human brains, even in this early stages. Imagine if we could also find a way to detect the inflammatory microglia in a human brain non-invasively and bring that together with some of the other biomarkers that already exist. To my knowledge, there's no way of doing that specifically right now. And that's one of the goals of the research that we're doing with Curals and a lot of our collaborators across the whole group. And I'm going to give you a couple of examples of that today. So really what we want to know is how do these cells change across disease progression, especially early, and can we develop new ways to both track them and target them early in disease? And I think the other thing to say about Alzheimer's disease, in contrast in some ways to other brain diseases, is one of the other reasons why a lot of scientists have become incredibly interested in microglia, not just because they're interesting cells, which is how I got interested in them years ago, but because human genetic studies, so GWAS studies and sequencing of the genomes of individuals that have Alzheimer's disease and risk of Alzheimer's disease, are more and more implicating microglia and immune system in this disease. In fact, many, many genes that have been identified as risk genes in late onset Alzheimer's disease are either expressed in or even enriched in microglia compared to other cells, including neurons. And for that reason, even scientists that have been studying Alzheimer's disease for decades started to pay attention to microglia. And that's led to a lot of amazing collaborations that I've been really fortunate enough to have across Alzheimer's disease, Cure Alzheimer's consortia and through the community of scientists that Curals is funding currently. And I just want to give you an example. It's not really meant to be looking at the individual genes. I just want to say that when you start to get data that are from sort of unbiased human genetics, that starts to tell us that, okay, these must be important, because if you have one or more of these genes, it's not like, one gene's going to tell you get Alzheimer's disease. But when you collectively start to see a lot of these show up, it's starting to highlight microglia or immune-related pathways that could lead to new therapeutic strategies. And I think this is a really important point. There are certain diseases like Huntington's where the causal gene has been identified. But even then, it's not clear that that might be the right target to go for. Whereas I think what this genetic studies are showing us, All the ones in blue here are saying, oh, these are genes or variants that are enriched in microglia. I think we can start to say, well, what is that telling us about the function? What do these genes do? How do they impact microglia? And how can we develop, as scientists, better models to really systematically test the functions? Because until we do that, we don't know how to then translate that into new drugs. And I think that's what a lot of the work over the last decade has been leading to. And I'm very excited to tell you about a couple examples of that. And one of the other things that have cracked open the field is development of new technologies that enables us to study microglia in an unbiased way. So it's really great to have hypotheses, but sometimes it's really good to let the data tell us what's important. And it's sort of the convergence of this genetic data and unbiased approaches that are starting to point to, the right pathways and targets to go after. And one of those technologies has been this technology called single cell sequencing. And I don't want to get into the detail here, but I just want to go back to these old images, not old, but like pathological images that after someone, passes away of diseases like Alzheimer's disease, we know, as I showed you in those earlier images, that microglia can be seen around plaques and around dying neurons. But, that's a snapshot in time, many, many years after someone's developed the disease. But because of this new technology, we can take those tissues, either from a mouse model or from a human brain, and we can break up the cells in the brain, and we can sequence their individual cells, and we can ask, what's different about the microglia that are around amyloid plaque from the microglia in a different part of the brain? Or how do those change at different stages of disease? And that's giving us new insight into what they might be doing. And I'll come back to that in a bit. And I also just wanted to say, these imaging experiments often tell us a lot about some of the functions that are really, really important. And as I mentioned at the beginning, one of the things these cells are really good at doing is engulfing or eating things. So we call this phagocytosis. Don't worry about the name, but they're, I would say another word for phagocytosis is sort of eating or engulfing. So when this neuron in the brain that I just showed you here is dying or damaged, do you see how quickly the microglia are pulling in and eating those damaged neurons? That's a really good function of microglia. But the other thing we noticed a number of years ago, they eat a lot of other things in the brain. And one of the things that we discovered, again, through my work in developmental neurobiology, is that they're also really good at engulfing synapses, which is the junction between neurons. And I'm going to give you an example of that. So in a brain, we have, billions of these synaptic connections, right? Even trillions, actually. And this is the point of communication between neurons. This is how neurons talk to each other. And why I'm telling you about this is because, without good communication between neurons, like this is how, we're able to do all the things we do. Like all of our cognitive functions are depending on the neurons talking to each other and rapidly communicating through each other at synapses. And what we noticed a number of years ago, and others in the field as well, is in addition to sensing damage and amyloid, look at how they're touching and interacting with these red little synapses. This is a red version of this from the mouse brain. And when we saw things like that, we also started wondering, could they also be nibbling off or engulfing synapses, right? Now, during development, that's a really good thing. It's part of the sculpting process that happens. And that's what I was studying in my lab. But we also know that synaptic loss, the loss of these connections, is a hallmark of many brain diseases, including Alzheimer's disease. And in Alzheimer's disease, there's evidence in human and in animal models that this can happen decades before you start to notice that you have Alzheimer's disease. In fact, it's one of the strongest correlates of cognitive decline is the synapse loss. So if we can figure out a way at the very earliest stages to identify when someone's synapses are vulnerable and prevent synapse loss, well, that might be a new therapeutic target. And that's what my lab's focused on for the last 10 or more years. And I'm going to tell you an example of not only some new insight, but also how some of that's already translating into new therapeutic approaches. So we want to protect our synapses, okay? And I'm not going to get into these other examples, but even though I'm going to focus on Alzheimer's disease, synapse loss is a hallmark of a lot of other brain diseases and disorders as well. So if we can discover how to protect our synapses in Alzheimer's disease through interaction with microglia, this might also have broader therapeutic implications for other diseases, because oftentimes when you have one of these diseases, like Alzheimer's, you can have co-pathology with other of these neurodegenerative diseases. So it's important to think about that as well. So we discovered, and I don't want to get into the details, into the like we say, the weeds here, but one of the things we noticed when microglia, I'm going to go back for a minute. One of the things we made an observation about is that microglia are nibbling off or eating some of these synapses in the normal, healthy, developing brain. So there's been no inflammation. This is just a normal function of these cells. And that led to this question: how do they know to engulf this synapse, for example, but not this one? Could there be sort of instructive cues that tell the microglia, oh, eliminate this synapse, but leave these other ones alone? And that's the basis of a lot of the research in my lab, is to try to understand those molecules and what they might be. And it turns out that some of the same molecules that microglia use to engulf or prune synapses are some of the same molecules in the immune system that one of those macrophages in your body use to recognize a pathogen, like a bacterial cell. And it turns out there are these groups of molecules called complement, in yellow here, that essentially tag bacteria and tell macrophages in the body, eat me, get rid of me. It's like our first line of defense against a bacterial infection. So this is an ancient way that we clear infections in our bodies, right? And what we discovered is that one of the things that happens in the brain is some of these same immune molecules are actually normally expressed in the brain. And instead of tagging bacteria in the normal healthy brain, some of those are tagging those little red synapses I just showed you. And that's telling the microglia, wow, eat me, remove me. This is just an example. So these complement molecules are tagging synapses. And what we discovered is that's a good thing during development, but it can be a detrimental or bad thing in the context of diseases like Alzheimer's disease or aging. And so the question is, does blocking this one way of pruning synapses, could that be protective in the context of Alzheimer's? Could that actually protect our synapses and reduce synapse loss? Now, of course, we really want to know the answer in people, but we can do these experiments in mice because we can block complement itself through lots of different tools, genetic and with like kind of blocking antibodies and other approaches. And we can simply, in these mouse models of Alzheimer's, ask if we treat mice with complement blocking antibodies or block this pathway, can we protect our synapses? And it turns out that there's a number of different companies like making tools to do this, including Annexon Biosciences that has developed an antibody that prevents this complement pathway from activating in the brain. So it's against this molecule here. And so we and others in the field, not just my lab, have used this antibody, treated these amyloid or Alzheimer's mouse models with this antibody, and simply ask, can we protect the synapse eating or the synapse loss? And through a number of different experiments from several different groups, including Genentech, it's a company that's been studying this, inhibiting the complement pathway prevented some of the synapse loss and also some of the cognitive decline, again, in an animal model, in a mouse model. And that's one of the first sort of proofs of concepts we would need to be able to then move forward into thinking about therapeutics. And of course, I'm not telling you this is the magic bullet or what it's going to, prevent all Alzheimer's disease, but I think it's an important example of how understanding a mechanism and uncovering, sort of normal, mechanisms in biology from the lab can then lead to collaborations with industry and the development of new approaches to potentially target this in patients. And in fact, that particular company, Annexon Biosciences, that is in the clinic in phase 2, not in Alzheimer's yet. They're doing this in other contexts, but already that's a promising sort of example of how we can sort of translate some of the work into the clinic. And I'd love to start to think about, could we start to, come up with other ways to do this in the context of Alzheimer's disease? And can we also start thinking about how to validate this in the human brain and human Alzheimer's disease? So we are starting to do some of that. And we're also interested in understanding whether complement could also be an early biomarker. Remember, I told you we want to kind of know whether our synapses are starting to become vulnerable or lost. We don't have a way of doing that right now, but at least through our work in other disease models and in other disease contexts like Huntington's disease, there's some promising preliminary data that elevations in complement is happening quite early. And so maybe it could be not only a target, but also an early biomarker for this pathway activation in the context of diseases like Alzheimer's. And this is work actively happening in my group and across many different groups as well. But complement is one of many, many targets. I just told you about the genetics that are implicating lots of other things that they do, other therapeutic strategies that we're all thinking about. But again, we need to start to think about how do we go about this, not just one molecule at a time, or going after one hypothesis at a time. That still has to happen. But we also want to be more kind of broad in our thinking, and through collaborations and through all of the research that's happening through the many different consortia and also through scientists across the Cure Alzheimer's scientific community, we're really starting to work together, because it really takes a village to tackle such a hard problem. And again, we're really trying to get at these early changes that are happening in the context of the disease. And so one approach, and I'd say this is one of the approaches we and many others are taking, is to start to measure changes in the cells that are changing across these diseases, not just in microglia in the brain, but also in the CSF and the blood of patients that we can collect across disease progression in these different cohorts. We can start to use the sort of unbiased, remember I mentioned single cell sequencing, we can kind of see which cells are changing and when, to identify disease-associated states that we might want to study more deeply. We want to leverage all of the genetic studies that are already implicating risk genes that reside in some of these cell states to prioritize which cells do we want to focus on to target. And also we need to develop better models that better capture the human states that we see in the patient. And that's what I want to end with right now, because I think animal models have been really powerful. We wouldn't be able to do any of the work I just told you about without mouse models that have sort of really been an incredible way of being able to control the situation and test hypotheses. But we also realize that the mouse and the human, very different, obviously. And what's really been exciting, I think, in the field is we now have the ability to take blood and generate from these blood cells what we call stem cells, and grow them into a dish and generate different cell types of interest, including the very microglia that I was just telling you about. So we can start to take blood samples from patients that have Alzheimer's or have increased risk of Alzheimer's versus controls. We can take and generate through a lot of different protocols that have been developed in the field. We can essentially make iPS or stem cells that look a lot like the microglia in our brains, and we can use those as models to study in a dish. We can start to systematically understand what changes their states and their functions, and then we can start to then go back and understand how we can then go back into the patient and go back into animal models to test their functions. So for example, in our iPS cultures of microglia, they're also very good in engulfing things like amyloid, synapses, and other substrates like tau. So imagine if we can systematically do CRISPR screens and drug screens to try to, in a much more high throughput way, develop those therapeutic targets that have been implicated by the human genetics. And that's exactly what we and others are starting to do. So it's quite exciting. And so the ultimate goal of the work I told you about today, that is really happening more and more across the world, laboratories are starting to do this. We find important signatures of disease from patient samples now. We can identify more predictive biomarkers that enables us to stratify patients, because Alzheimer's might be different depending upon the stage of Alzheimer's or the type of disease you have. So we want to be able to identify early, stratify patients in a way that can lead us to predict who's going to go on and develop these diseases. And we really want to start to bring the immune and microglia components into the biomarker space. This is something that we're really working hard on doing. And then from them, we want to build better models and better platforms to test some of the strongest leads that emerge from this work. And that is a way of translating the work and building better partnerships with industry and others that are in that space to, as a sort of community, start to scale this work up and to develop drugs faster. So I think this really, to me, illustrates the power of what Cure Alzheimer's Fund can do, because it really does take a village. All of the scientists that are working in this field now have been working very hard. But one of the things that Cure Alzheimer's Fund has done and enabled and catalyzed, in my opinion, is truly collaborative science, where we don't work in silos. We don't just do the work in our lab and not share the results. We're in real time collaborating, sharing ideas, sharing data. And that is enabling us to do this together because it's such a big problem. And that's where the consortia that Cure Alzheimer's Fund has also been so generously funding, because it enables us to bring teams of scientists together. And in fact, I lead one of these consortia focused on microglia and neuroimmune and neuroinflammation, so that we can really, across our labs, our collective expertise is enabling us to start to do this much faster than any one lab or any one scientist could ever do alone. And I think that, to me, is how we move the dial faster. There's been incredible progress, in part due to a lot of the support from Curals, but there's so much more we need to do. As we learn more, the more we need to delve in and sort of understand the biology deeper. So that's going to require more and more of this sort of collaborative team science. And so I want to just sort of, end by giving you sort of an example of some of the things that we've been doing. But I also want to say, not only does it take a village, all of the trainees that work in our labs, these are the students and the postdocs and the graduate students. These are all the people in all of our labs that make it all work. This is what we call the human microglia. I'm in the middle, and these are like the branches of a microglia. But you can imagine we all have to work together to be able to make this all happen. And I think one of the other things that I have come to appreciate, and also Cure Alzheimer's comes to appreciate, is we're also training the next generation of scientists. We want more and more scientists to be in the field doing this together. And that is another one of our major jobs as scientists. It's not just to generate amazing data and make discoveries, but we got to train all these guys to work together and lead the charge forward over the next decade and more.